Welcome to Ask the Expert. Today, we are very pleased to have with us Dr. Kalpana Mandal. She's a postdoctoral researcher at the Terasaki Institute in Los Angeles, and her work centers around oxygen-generating microparticles for ischemic injury. Really interesting. I'm very interested to see how what she's thinking about or where she is in this technology development, and then what she's thinking about, like whether or not this might even be applicable to eyelid implantation and the like. So I'm really interested to hear about this, Dr. Mandel. Thank you so much for joining us. Do you want to give us a quick brief bio?
Thank you. Thank you so much for the opportunity, Monica. And I am really delighted today to talk here and looking forward to the collaboration and the opportunity to use my technology in diabetic patients. If it can be transplanted, it will be great. So I will show today what I'm going to do today as it is. So what my background is. So first of all, I don't come from a medical background. So I did my bachelor's and master's in physics. But then I was very motivated to do biophysics. So during my undergrad training, I was doing some biophysics project with cancer research, and that motivated me to move towards biophysics research. And then I moved to France for my PhD, where I worked on the role of extracellular matrix, how it is regulating intercellular organization and force generation. But that was more fundamental question and applying physics in biological system. But I wanted more in the medical side, how I can use my technique and knowledge to improve and healthcare spectrum. So then I started working in the biomedical field, and in past I have worked in liver cancer research, where I was working to develop and understand different biomaterials and how the mechanical properties of the biomaterials are regulating the force generation or the cell function. So
Yeah,
that was in past, yeah.
No, that's fantastic. It sounds like you have sort of excellent training to kind of, address these questions.
Yeah, so I am happy to work at the interface, and I think I really enjoy this kind of environment where I can work with medical doctors, engineers, physicists, biologists, or bioinformatician. So I think those sort of environment helps to solve a problem very quickly.
Yeah, I agree. And
And
is this the case for the Terasaki Institute for Biomedical Innovation? Do you feel it's a very collaborative space there in Los Angeles?
yes, I think that this institute, though it's this direction is new, the one we are working right now, but I think it has very great future because it is very focused on transplantation, and it has different departments where there are biomaterials research, then cancer immunoengineering, and then wearable devices and electronics. So different kind of aspects, and there are doctors as well, and the collaboration with academia and industry is very strong. There are a lot of funded projects from industry, which is really helping us to see and visualize how a product which we are doing in the laboratory can be translated and could be improved for humankind. So I think it is very great opportunity for us for this institute. And
Yeah. No, I agree. And we actually interviewed Dr. Ali Karamasani a year and a half ago, I guess, realizing that he came from MIT and he had this really great vision of wedding
yes.
both, the academic research and industry. And it looks like you're at the forefront of that. So I cannot wait to hear more about your work.
Thank you so much. Yeah, it's a very, very great opportunity for us. Yeah. So without any delay, I will go to my next slide where I say the islet transplantation. So the major problem here is with type 1 diabetes that the patient's immune system is destroying their islet cells. And to improve that, so there are islet transplantation, people are working on that. But that is also not happening because the immune system again is destroying. So what people do is, so the researchers, they have implanted the cells in a polymeric environment so that it cannot be destroyed easily until it's vascularized and everything is set there, implanted properly. But in that case, the main reason is that when it is inside the polymeric network in a mesh, the supply of nutrients and oxygen is very limited, and then cells die. So here, I think that this is not only in case of diabetic patients, because this is a major problem when there is a tissue implant, engineered tissue implanted in the body, and the vascularization is a major issue, because that is in case of if there is no vascularized network, nutrient supply and the oxygen supply will be limited, and in the big tissue construct, it's more problem. So I will discuss more. So here, in case of islet transplant, what happens? There are no vascularized network where they can get the oxygen supply in this transplanted tissues. So these problems I try to address in my research. And then not only in this, at the single cell level, what happens when there is no oxygen supply? So at the single cell levels, you can see that there is a much more ROS production. There is upregulation of metaphagy. And then there are hypoxia inducible factor, which is like constantly degraded in normal condition. But if there is no oxygen supply, it upregulates. And there is apoptosis, cell death in the implanted tissue. So there are a major challenge in the tissue engineering field with this supply of oxygen, and this hypoxic core occurs in the tissue implant. So another issue with this is that when there is hyperglycemia, so there is a cardiac insulin resistant, and then there is also oxidative stress, and you can see that there is microvascular dysfunction as well, and which leads to heart failure in the end, because there are other things happening, like such as fibrosis, cardiac stiffness, and cardiac diastolic and systolic dysfunction. So there are major problem occurs when there is hypoxia in case of hyperglycemia as well. And then when the heart failure happens, and during the transplantation, what happens? So the heart can be preserved in the window of 4 hours roughly, but then the ischemic injury and the ischemic time is prolonged, and then this allograft transplantation, and that is rejected by the patient and increases the mortality. So in 2017 report, there are 8,000 hearts are wasted, and approximately 20 deaths per day because of the delay of organ transplantation, because then if it is delayed, and then it will be more ischemic condition for prolonged time. And so for all these reasons, so as we see that there are major challenges with the oxygen supply, so we come up with a solution where we can supply the oxygen in a given in a particular way, because you cannot perfuse the gas in a large amount, because you need, like for a let's say for in case of islet transplantation, you need 100 Hg oxygen for that, but you cannot just with a gas supply, you cannot do that. So you really need to come up with a solution where you can supply oxygen in a sustained manner and enough amount that the cells does not die and it gets enough amount of oxygen.
Yeah, very tricky balance.
Yes, it is very tricky balance. Yes. And the sustained amount of oxygen is, sustaining the amount is very important here, because you can supply the oxygen with different ways, but in sustained manner, it is the important here, because if it's too much oxygen, then also cell will die. There will be effect on the cells as well. So this balance is very important. So here we are trying to solve this problem and increasing the tissue functionality post-transplantation or or after organ transplantation, any kind of either tissue or organs such as heart. So here I will show how we are working with the microparticles. So previously we have done oxygen releasing biomaterials. So where we have used calcium peroxide as a powder. And when we put it in a polymer matrix, what happens, this calcium, when a calcium peroxide, when it is in touch with like media, it has a burst release. It has too much oxygen. So it's not a sustained manner they can release. So then we came up with an idea that if we can encapsulate them in a polymer, then we can see if it's releasing in at a certain rate, which we are expecting for in vivo, implantation. So yes, the way we have done this, so we created oxygen releasing particles with calcium peroxide using PLGA polymer. And then we try to see, as I said, that in case of this hyperglycemia, there is a cardiac stiffness. And then we try to see how the contractility is affecting when we are adding this particle. And we try to measure the forces and try to see the functionality before implantation. And then we tried in rabbit hearts for the transplantation, and then see whether how the ischemic injury is improved with our particles. So this is the outline of the talk. So I will go maybe a little quickly. How much time I have? Okay, I have 15 minutes.
Yeah, it's fine if we go a little over. It's totally fine.
Okay.
I really just the main thing is we want to,
See.
really hear your story.
Okay. Thank you so much. Yeah. So, then we prepared this particle without using water, because that will decompose the reagents immediately into oxygen. So we don't want that. So we prepared with immersion method in an organic solvent, and then we prepared different concentration of particles here. As you can see, that CPO PLGA ratio you can see here, just to optimize, and the main goal is to achieve a sustained amount of oxygen as like in vivo. So we tried these 3 different concentration, and then the way it will work, so we have PLGA and Ca2, which is mixed in the emulsion method, and then when this is in contact with water, it starts decomposing like calcium hydroxide and hydrogen peroxide. But then in the solution, if we have catalyst, then this H2O2 breaks this, this catalyst breaks the hydrogen peroxide into oxygen and water. So this is the main mechanism.
Oh, sorry. Can I just ask one quick question?
Sure.
So is this microparticle, fine with being implanted in vivo? I mean, what's the level of toxicity? I guess I want to ask how safe is it to implant in the body?
Yeah, so PLGA, the polymer we use, it's FDA approved polymer. So it's really safe for transplantation. And that is the main reason why we have used PLGA, that it's easy, which is already approved, easy to, transplant. Perfect. So that is the main reason. And I will show you the toxicity experiment data, how it is improving the toxicity in the later slide.
Fantastic.
So, yeah, so these are the particles which we have prepared, and this is the empty without any, calcium peroxide. And you can see that when we introduce that calcium peroxide into the particle, there are homogeneity. And if we zoom more, there are very porous structure inside it, but it's very few. But the main idea is that when it is in contact with water, it creates the pore because it's breakdown and it decomposes. And as much as it's going inside, and it's trying to create the whole pores inside these particles. And also why we measure the size, because the size also is important factor in the releasing profile. So if it is a bit higher bigger particle, it's the releasing profile will be different than a smaller particle. So we see that in this method, it is in a comparable range. But this method could be improved, which we have not done here, by microfluidics, where you can create the particle in a given size. There will not be any distribution at all. So which we are working on right now to improve this for these particles. So next, we try to check that whether this calcium peroxide is encapsulated or not. To confirm that, we did the alizarin red staining, which binds to calcium only. And you can see the calcium red, alizarin red staining here, where confirms the calcium and encapsulation. And we measured the zeta potential, but what it says is that the stability of your particle, like whether it will be, clamped or it will be like separated, how stable they are, so that it says, and which is in the range of, stability range below minus 20. So the range is 10 to minus 20 millivolt. So this is in a given range where the particles are stable. So after characterizing this, we would like to know the oxygen release profile for all these particles. So for that, what we did, we put these particles in a hypoxic solution and put them in a hypoxic chamber, so that whatever the oxygen we are measuring for each particle is coming only from the particles. So when we do that, so you see that the free CPO, which we did earlier, which releases the oxygen and it has a burst release, and then it goes down within 6 hours to 8 hours. But then in case of all these CPO 1, 2, and 3 particles, sorry, this is CPO 1, this is 2, and this is 3.
Right.
So with different concentration, you can see that we can achieve with 1 mg per ml particle concentration. It is in the physiological range, 21% up to 24 hours. And then we need to measure more. However, here I would like to mention is that this profile can be tuned by tuning the polymer, PLGA. So if you tune the polymer mixture, and then with that, you can, with different kind of PLGA, you can tune this particle release profile up to several days, like maybe 14 days or 21 days. So here I am showing the proof of concept here.
Yes, exactly.
So here, so then the experiment we did is with primary racked cardiomyocytes, the cardiac cells. So first we isolated the cardiomyocytes, then we cultured them for 2 days to have a monolayer, and the cell starts beating on the in vitro culture. And then we treat them with hypoxia, 1% oxygen and 5% CO2 in this chamber. After 2 days of hypoxic treatment, then we add the oxygen releasing particle in. And then we perform any kind of assays, which I will be describing now on. And here you can see that in hypoxic control, there are more cell death. But when there is particles, we are using 1 mg per ml and 2 mg per ml. And we see that there are decrease in cell death, and which you can see in the metabolic activity, that cells are very fine and their metabolic activity is increased, which was decreased in hypoxic situation. Then we tried with different cells as well, just to confirm these results, and we found the same results for the another type of cell, Hypoxia cells, which confirms our results for the primary cardiomyocytes. So coming back to your question, previously you asked whether this is how toxic they are. So they are quite fine. And another thing, a drawback I would mention in the end, or maybe now, maybe I can say, because with this calcium CPO particles, the pH changes during this time period because there will be free radicals. But that can be controlled by using Hepes buffer. So when we use the Hepes buffer, we showed that, I'm not showing the data here, but we showed that pH can be controlled with adding the Hepes buffer to the media. So next, what we did is that we checked as I said, that this hypoxia inducible factor, which is upregulated in when the cells are in the hypoxic situation. And then we showed that when they are in hypoxia, whether they are upregulated or not. And we see that in this fluorescence image, they are upregulated. And then with the CPO particle, you can see that these hypoxia inducible factors, they are getting downregulated. But what we see here is that the immunofluorescence images. But to assess more carefully, we need to show it in protein expression. So we are working on it with Western blot and qPCR, and the Western blot result, which I'm not showing here, which also shows that the hypoxia inducible factor is in increases, and then with our CPO particle, we can downregulate that expression when we supply the oxygen. So the next, I show you here the effect of beating.
Wow, this is a very a fascinating result.
Yeah, this is really good result. I am really happy with this microparticle work. And you can see that it's very clearly visible. But oops, what happened? Oh,
maybe just put the slide back up.
Oh, somehow my PowerPoint.
Right when we were at the good part.
Yes.
No worries. Just try to, put it back up and we'll just continue. It's not a big deal.
Okay, so. Okay.
Here we go.
Okay. Yeah.
Good review of the protocols. There we are.
Yes. So here we were, where I was showing you the beating. Yeah, so as you see in this hypoxia, the beating is very, slow. And here you can see that we could improve the cardiac breathing with this oxygen releasing particle. And I will show you the quantification. Oh, I think it's because of the video.
I think it is. Yeah, it's the video, but that's okay. We can try.
Yeah, I already have shown. It's fine. So I will go directly to the next slide.
Okay, perfect. Who knew that the video would mess everything up? Even though it is a beautiful, slide, and some really, it really shows quite a difference just visually.
Visual, yeah, exactly.
And you were
without quantification
to back it up quantitatively.
Yeah, exactly. So here is the quantitative data where we calculated this beating, and you can see that we can improve the amplitude and the frequency both when we are adding this oxygen releasing particle to the system.
Nice.
And here you can see the quantification. So with that, I would like to go back to the introduction slide where I showed that the cardiac stiffness and dysfunctionality in diastolic and systolic function. So I think that those are the cardiac functionality could be improved in using these CPO particles, and during the heart transplantation also these particles could be used, I think, altogether. But then since I am from physics background and I want to quantify things more, and here I wanted to quantify the tissue contractility and how much I am improving the contractility by using this particle. So what we use here, we use the chip where we design the cardiac tissue on this micropillars. As you can see that there is a pillar, and we design this microchip in PDMS. And then when the tissue is formed, we created the tissue, the primary rat cardiomyocyte tissue in between these pillars. You can see here, as you can see. So when this tissue is contracting, and you can see that this pillar is getting deflected. And from this deflection, one can calculate very easily the contractility of the tissue. And here you can see the different staining of nucleus, actin, and the hypoxia inducible factor. All are stained here, and you can see that the tissue is surviving and contracting very nicely. So when we do the quantification, and you see that the displacement in case of hypoxia is very less, whereas when we add the CPO particle, we can improve it, the displacement. And then when we can calculate the force or the contractility, and you can see that there is a significant amount of increase in the measured force in this system. And the next thing, so here I showed you like contractility for 2D monolayer, the cardiac beating, and then the 3D cardiac tissue. But then we also have done the force measurement for a single cell level. So the way the contractility or the force is measured for the single cell, so you have a very soft substrate here where there are small fluorescent particles. These particles are different than CPO particle. So this fluorescent particle, when this cells are on this soft substrate, and then they are exerting forces, or they are contracting, they are deforming this substrate. And when they are deforming this substrate, this fluorescent particle are displaced. So from the image where you kill the cells, where the particles are relaxed, and when they are contracting, so when they have a stress applied on these particles, you can obtain a displacement field from this two, so which you can see here. So this is a cell, and this is the displacement field for this small fluorescent particle. And from this displacement field, you can calculate the force exerted by the cell, or the contractile energy is generated by the cell. So the way it is being calculated, I will go quickly to instead of explaining these equations. But the way it is done is you have a displacement field, which is denoted by U, and you have a force F. And the G is the Green's tensor, which is related to the substrate property. So this is known, and you prepare the substrate with a given property, and then when you calculate this displacement field, as I showed, and you can do fast Fourier transform mathematical calculation just to obtain this force. And when you do that, and you can see that for this cell here, I am calculating, this is the small particles, and I can calculate the force for this particle, for this cell. And when I do for many cells, and I average them for hypoxic condition, and when I treat them with our oxygen generating microparticles, and you can see that the stress generated by this cell could be improved with this. And the generated energy by the cells on this substrate is also higher when CPO particles are added. However, the cell areas are same for both these two cases. Then we move to the next part in animal studies, where we wanted to see the effect on heart in a rodent model, and we used rabbit hearts. And after procurement, we have put them in a regular normal saline solution. And then we have formulated our media, which is adapted from Celsius, which is one of the organ preservation solution. And with that adapted media, we added our CPO particle. And you can see that in this saline condition, there are a lot of ischemic injury in the tissue. Whereas when we do that, adding the CPO particle and in our formulated media, we have less ischemic injury in the tissue, which is quantified by the waviness of myocardial fibers and myocyte hypereosinophilia. So on that note, I would like to thank all the people who contributed to this work, and the grants. It's not my grants though, but this project is funded by these grants. And also the heart cells, rat heart cells, we got it from the UCLA NRVM hardcore. And thank you for your attention and giving the opportunity to talk. And I hope that there will be a lot of collaboration and interest in this system.
No, it's a fascinating system, Kalpana. And I, obviously it's like, your oxygen generating microparticles are creating a better behavior in these cardiac myocytes. And
Yes.
can you just, just quantitatively and qualitatively, so that's seen very clearly. I wondered if you might comment a little bit about this system and how it might have, oh, bless you, and how it might have some bearing on islet implantation. As we know, one of the biggest issues for islets when they're implanted in vivo is they need oxygen. And they need to get oxygenated to function properly. At the same time, they need to be protected from immune assault if they're getting implanted into someone who already has an autoimmune repertoire ready to attack. So what are your thoughts around this? Can you expand that?
Yeah, I think that this would be very great opportunity to expand my work in this direction. And I think that with while encapsulating this islet cells in the inside a polymer, I think that the particles which size can be controlled, and we can prepare in a nano formulation as well, and which can be encapsulated in a 3D printed in a manner where the particles and the cells will be inside, and then can be transplanted, I think, the in vivo, and that way it can have the sustained release for a certain means, depending upon how long we want it, we can tune the particle oxygen release profile, and we can 3D print them. And I think that will be one of the solution, I think, could be used in this scenario.
Yeah, it's very interesting. And I would encourage those who are deep into this technology, maybe ViaCyte, maybe down in San Diego, maybe Vertex in Boston, to kind of think about reaching out to you, maybe a second postdoc, or maybe a position at industry like that would be really exciting collaboration and opportunity. I think we spoke to Nayara Leite yesterday. She is working, she came out of the Melton lab at Harvard, and she's currently working at Vertex. And that is, they're really approaching this strategically. So I hope that, people will listen to this, hear it, reach out to you, and, further the scientific collaboration and accelerate the work. Thank you so much for talking to us. It was great, beautiful work. And I look forward to speaking to you again in the future.
Thank you so much, and I really appreciate this opportunity to talk, and I hope that many people reaches out to me, and we can move forward to solve this problem in the diabetic landscape.
Thank you so much. Have a great rest of your day.
You too. Thank you so much. I appreciate it. Bye.